Good afternoon, folks. Hope everybody is doing okay today. This is going to be my lesson on the first lesson, maybe one, maybe two. We'll see how this goes, on mutation and cancer. If you have not already watched my lessons on DNA and RNA, you probably want to go back and do that because then you'll have the necessary background you need to understand what I'm talking about here. And if you are in my biology class, you should probably go on Blackboard and get a copy of the handout for this lesson because some of the things I'm going to be describing will go a lot better if you have a visual to represent them. Now, in my last couple of lessons, we've talked a lot about genetic information. We've looked at the DNA, the chemical structure of that molecule, and we've seen how it replicates itself when cells divide, as well as how it records biological information in the form of genes. Then we took a look at RNA, another related molecule, and demonstrated how RNA acts as the bridge between DNA and the various protein molecules that your body knows how to synthesize. That DNA is a recipe book for proteins, and RNA is the intermediary molecule that helps it to execute its instructions through the process we know as gene expression. Now, everything that I talked about in the course of all this is reliant on having a set of standards in which DNA is faithfully copied. Now, you saw back when we discussed DNA replication that there are a lot of mechanisms working to ensure that when we copy the DNA molecule, nothing goes wrong. First off, the complementarity rules of the two strands, A always goes with T, C always goes with G, means that it's hard to make errors in the first place. Wherever there's an A on one side of the DNA molecule, there's got to be a T on the other because nothing else will form a proper hydrogen bond with it. That's one big defense. We also, of course, have the proofreading function of DNA, which makes sure that if we do make errors in the process of replication, they get caught. So there are a couple of things going on that are helping DNA to maintain its faithful structure. And for the most part, you don't have a lot of errors in DNA replication. The molecule tends to retain its integrity across biological time. There are genes that you have that are exactly the same as the genes in every other human being. And there are genes that you have that are exactly the same as certain genes in every other vertebrate, every other animal, indeed in every other eukaryote. You share about 33% of your DNA in common with bananas. And that means that those genes have pretty much stayed the same across time. They have not really been modified over the course of biological history in any significant way. Now, the thing to remember, however, is that this process, like no process is perfect, right? And that includes DNA replication. That even though there are things that are there as fail-safes to help protect against errors in the process, it's not foolproof. Your DNA is 5 billion base pairs long or thereabouts. That's a lot of room for an error or two to creep in, even with defensive mechanisms in place. And so that gets us to our point today, the discussion of a thing called mutation. M-U-T-A-T-I-O-N. Mutation. Now, you've all heard the word mutation, and when the word mutation comes to mind, or the word mutant comes to mind, M-U-T-A-N-T, a mutant is any cell or organism that has a mutation. When you hear the word mutation, the things that tend to come to mind are, giant movie monsters that eat Tokyo, or dudes with superpowers. And I'm here to tell you that, unfortunately, these are probably not the outcomes that are most likely. Well, okay, maybe that's actually fortunate given the way that things tend to go for big cities and superhero movies, but you know what I mean. Point is, mutation as a phenomenon is generally a lot less dramatic than what you're used to seeing portrayed in fiction. I should also like to point out that the word mutation, as we use it in biology, is generally value neutral. In other words, mutation does not automatically connote a good thing or a bad thing. What a mutation refers to is any change in the sequence of DNA. That's a mutation. Any change in the sequence of DNA. In other words, the DNA molecule has been modified in some fashion from its original form. It no longer contains the exact code that it started with. The likeliest time for mutations to occur is during the process of DNA replication. Whenever the strands are unzipped from one another and you're building new strands onto them, that's the most vulnerable time for the DNA, and therefore the moment when it is most likely that something is going to affect it and cause a change. The other time that mutation is fairly likely is during mitosis or meiosis, because during those times, the DNA, at least in eukaryotes, no longer benefits from the protection of a nucleus, and therefore is more subject to damage from outside forces. Furthermore, if it's happening right at that moment, it means that we're potentially creating a new cell that has that mutant gene in it from the start. So when a mutation occurs, the sequence of the DNA has been changed. It is no longer going to be as exactly what it was that you started with. And what I want to talk about today is how that can happen, what effects it has on the DNA molecule, and what the consequences for you, the organism, will be. So starters, how does this occur? We've already established that there are a lot of fail-safes to protect DNA against random error. So how do we get around them? A couple of things here. First and foremost, mutation can be caused by chemical interference. DNA is a chemical, it's a molecule, it's a nucleic acid, one of our 4 basic biomolecules. And like any molecule, its reactions can potentially be disrupted by outside forces, whether that means energy, whether that means chemical interference, one way or another. But we're talking specifically about chemical interference right now. You know that your organic molecules are, for the most part, fairly delicate things. They rely on very precise conditions of temperature, pH, and other features of their chemical environment in order to remain intact. If you interfere with those conditions, the molecule in question can be damaged or even broken down altogether. It is possible that the DNA molecule can be damaged by outside chemical exposure. Any chemical that is capable of causing damage to the chemical structure of DNA is known as a mutagen. M-U-A-T, sorry, not mutagen, no, that's right. M-U-T-A-G-E-N. M-U-T-A-G-E-N, a mutagen. So mutagens are any chemicals potentially can cause mutations. Sometimes that's caused by disrupting the chemical process of mitosis or meiosis, interfering with what's going on at that time in a way that damages the chromosomes. Sometimes that's involving a chemical that's going to interfere with the process of DNA replication, with one or more of the enzymes or accessory proteins involved in replication being disrupted in its activity. Sometimes the mutagen just directly damages the DNA molecule itself. But whatever's happening, a mutagen is any kind of chemical interference that results in DNA having a change in its structure, either because its process of formation or division was disrupted or because the molecule itself was directly attacked by an outside force. There are a lot of mutagenic chemicals. A lot of things are mutagenic in high doses that are not in smaller doses. And there are things we simply don't know if they're mutagenic or not. We are aware of certain mutagenic chemicals. For example, benzene, for example, is a well-known mutagen. There's a subfamily of mutagens that bears mention. A group of mutagens called carcinogens. C-A-R-C-I-N-O-G-E-N-S. Carcinogens. A carcinogen, whenever something is carcinogenic, that means cancer-making. Carcin or carcino means having to do with cancer, and genic or gene to create, to generate. So carcinogens are mutagens that specifically tend to cause cancer. They specifically lead to cancerous mutations. And we'll talk more about what makes a mutation cancerous a little bit further down the line here. So all carcinogens are mutagens. Not all mutagens are carcinogens. You're probably aware, at least I hope you're aware at any point, that a lot of the chemicals found in cigarette smoke are carcinogenic and are well known to have cancer-causing effects. So chemical interference, that's one possible way of getting mutations. Another well-known method by which mutations can happen is via the action of radiation. Radioactivity interfering with the chemical structure of DNA. Now the word radiation, like the word mutation, is itself kind of a loaded word. We hear radiation and we think atomic bombs going off or dangerous levels of radiation being emitted by a nuclear power plant or radioactive waste or something like that. The word radiation, I should point out, in physics means something much more broad-based. that radiation refers to any energy that travels in the form of a wave. Electromagnetic radiation is a better way of putting it. And there are a lot of forms of radiation that you encounter every single day, most of which are completely harmless to you. Exhibit A, one that you're experiencing right at this moment. Light. Light is a form of radiation. It happens to be the portion of the electromagnetic spectrum that is visible to the naked eye, that our eyes are capable of detecting. Anything between a wavelength of about 400 and 700 nanometers contains all the colors of the visible spectrum. Anything with a longer wavelength is called infrared radiation. INFRARED. Infrared means below red. Red is the lowest energy color in the electromagnetic spectrum that we can see. Anything that is above violet in the ultramagnetic spectrum, anything with a shorter wavelength than violet, is ultraviolet, ULTRA, violet. So be careful with the spelling. It is violet, VIOLET, not violet, VIOLENT. But so infrared radiation is radiation that has a longer wavelength and lower frequency than visible light. Anything that is ultraviolet has a shorter wavelength and higher frequency than visible light. The danger point with radiation typically comes with the high frequency forms. The low frequency forms are not a danger to you because they don't contain enough energy to disrupt your DNA. They simply are too low energy bearing to really cause meaningful effects. Like radio waves, for example, are a form of infrared radiation, but their energy level is so low that they can pass right through your body without any problem whatsoever. That said, anything higher in frequency than violet light, anything with a wavelength below 400 nanometers, shorter wavelength means higher frequency, carries more energy. The shorter your wavelength, the more energy the radiation is carrying. And anything that is ultraviolet, that's a dangerous level of radiation. That's where we need to start getting concerned. That's the level of radiation where you're likely to suffer a problem. The issue here comes because you've got extra energy disrupting the cellular environment. And you all know if you add energy to a system, that can potentially set off a chemical reaction that you weren't planning on. It can provide activation energy to catalyze a chemical reaction that was otherwise not going to happen. So high frequency radiation adds energy to a system, and when that system is a cell, it can potentially cause disruptive chemical reactions that might damage your DNA. Forms of radiation that can do this, most common one you might be exposed to in day-to-day life is going to be ultraviolet radiation, UV rays. You all have heard of UV rays because UV rays come right with the sun. Anytime you were out in the sun, you were getting UV exposure. Now, most of the UV rays that come with sunlight are filtered out by our planet's atmosphere. Most of them don't make it all the way to Earth. The lower the sunlight levels, the less they are happening. The more intense the sunlight, the more they're happening. That means the closer to the equator you get, the more intense UV radiation that you tend to get, whereas further and further north and south, the less the Earth is receiving that radiation from the sun. Obviously brighter in summer than winter. Where the planet is tilted in such a way that we're receiving a higher intensity of sunlight during the summer hours, and therefore you all know you're more likely to get a sunburn during the summer than you are during the winter. Because that's what a sunburn is, folks. A sunburn is a radiation burn. That is your skin being damaged by the UV rays. Now, part of our body's natural defense against radiation is having a thick layer of dead cells on the uppermost surface of our skin. All of this that I just ran my nails along, the tracks there in my arm where maybe you can't see that, where I just dragged my nails along myself, I can do that because that's all dead cells. You've got between 20 and 50 layers of dead cells present on the surface of your skin all over your body, depending on where in the body you are, more or less. And that layer of many, many dead cells is the primary screen against ultraviolet radiation. But it can still be damaged, and sometimes the radiation can break through the tissue underneath. And if you get a lot of ultraviolet radiation, the danger of mutation goes up. There is a reason skin cancer is the most common cause of cancer, because the most common type of cancer, I should say, because your skin is the part of your body that gets the most radiation exposure. And by the way, our natural pigmentation is part of radiation shielding as well. You go out in the sun, what happens? You begin to get a tan, right? getting the tan is basically your body saying, uh-oh, lots of radiation out here. I'd better add more pigment to soak it up. And the pigment in our skin, the melanin, is primarily an anti-radiation screen. So most people, when they spend some time in the sun, will get a bit darker within the limits of their particular genetic complexion. Not everybody can get equally dark. Some people have a baseline complexion that is naturally darker or paler. You might only get to a certain point where you just don't get any darker and you sunburn very easily. You might be very dark to begin with, and you don't see much of a change when you go out in the sun. If you map human skin colors on a population, like on a map of the planet, you'll see that the closer you get to the equator, the darker skinned people tend to be. And the reason for that is quite simply because that is an effective defense against very brilliant sunlight. All right, so other forms of radiation that cause you a problem? X-rays. X-rays are an even more intense form of radiation than our ultraviolets. You might have noticed whenever you get an X-ray, the X-ray technician, leaves the room when you're getting the X-ray, and you might think to yourself, "Holy crap, is this that bad?" A short burst of X-rays you're fine with. Frequent exposure is a problem, which is why that technician who has to give 30 of those a day leaves the room when you're getting it to protect themselves against that exposure. In the early days of radiation science, before we knew about the danger, X-ray shielding was not really a big thing, and a lot of people who worked with X-rays regularly succumbed to cancer. A very famous example, the woman who discovered radioactivity, one of the great scientists of the early 20th century. You might have heard of her, Marie Curie. She was one of the first women to really make inroads in the sciences, and to this day, she's the only person, male or female, to get the Nobel Prize in both physics and chemistry. Marie Curie discovered radioactivity in the late 19th century and worked with X-rays and radioactive elements throughout her entire career, and she eventually succumbed to a rare form of aplastic anemia and leukemia, almost certainly a result of unshielded radiation exposure. And the most powerful form of radiation, of course, is gamma rays. Gamma rays are the lowest frequency, highest energy form of radiation. Odds are you will never be exposed to a significant amount of gamma rays in your lifetime. In fact, you'll probably never be exposed to much by way of gamma radiation at all, because it's mostly screened out by our atmosphere. X-rays and gamma rays don't really make their way to our planet's surface with the energy of the sun. So unless you're working in a high radiation field, odds are you're not going to be exposed to any significant quantity of those. You might get some X-rays as part of like a screening, for a medical test, or like sometimes some airports still use X-ray scanners to look at people, although most of the new magnetic wave scanners nowadays. But unless you're an X-ray tech or getting lots and lots of medical testing, odds are you're never going to be having to worry about your X-ray exposure. Likewise, gamma radiation, you're probably never going to be exposed to any significant amount of gamma radiation at all unless it's part of a medical treatment. So you might have had or known somebody who had a radiation stress test, where what they do is they inject you with a wee tiny amount of a fast decaying radioisotope. It breaks down swiftly in your bloodstream, so it ceases to be a danger to you. But while it's circulating, it gives off gamma rays, and we can track the radiation coming from your body while that's happening, so that we can, say, map out your blood vessels and get an idea where there was damage from a stroke, for example, or where there might be a tumor blockage or that sort of thing. Also, if you do have a cancer, when you get radiation therapy for cancer, that is exposing it directly to gamma radiation. And yes, there is a paradox here in that radiation can cause cancers, but radiation can also kill cancers, because most cells, including cancer cells, will be killed dead when you expose them to intense enough radiation for long. And by the way, in terms of sources of radiation in your day-to-day environment, you do not need to worry about radiation from a nuclear power plant. The shielding around them is so intense that you could pretty much be leaning against the outside of one while it's operating and not be exposed to any more radiation than your television is emitting. Yes, your electronic devices emit small amounts of radiation, but it is not a threat to you. There was a big controversy a few years back in which everybody was worried about... Is there a light blinking on my phone? Huh, okay. Everybody was worried about your cell phones giving off radiation. And there was this big scare for a while where people said, oh, cell phones are producing lots of radiation. Well, I'm here to tell you that your cell phone does in fact use radiation to send signals, but that form of radiation is what is called microwaves, and microwaves have even lower frequency than infrared radiation. And as such, there's zero danger. Anybody who tells you that the radiation emitted by your cell phone is a danger to your well-being simply does not understand physics. And finally, in terms of causes of radiation, radiation's around, it's in our environment, you get bombarded with it every day. One last thing I wanted to mention about that, tanning bed, do not tan yourself. Do not get into one of those things. That is a cancer machine. You're exposing yourself to levels of ultraviolet radiation that you would normally not get outside because the atmosphere filters them out. So don't do that. It's going to potentially give you much, much higher risk of skin cancer, and you're going to end up looking a funny color like Donald Trump anyway, and you really don't need that. All right, so next possible cause. You might not think of this as a cause of mutation, but it's a thing that you do have to know about, and that is viruses. I think I mentioned in one of my previous lessons that certain viruses have a method of reproduction where what they do is that rather than just invading your cell and like taking over the cell, some viruses have a much more subtle method of getting themselves copied, where they move into a living cell and they attach themselves to its DNA. They basically latch onto your DNA, effectively become part of your DNA, and then they just get replicated along with the rest of the DNA whenever the DNA undergoes replication. You actually have more viral genes riding along in your DNA than you have human genes in your DNA. And most of them are completely harmless. They're just there. They're just viruses that don't have any harmful effect whatsoever that sort of latched onto your genome possibly millions of years ago and have just been getting copied silently ever since. We believe that certain viral genes may have actually triggered important events in evolution. For example, it's thought that viral genes may have played a role in the evolution of sexual reproduction from asexual reproduction in the first place. But viruses are going to be an interesting thing here because when that virus attaches to your DNA, A, it's adding new genes to your DNA, viral genes, and B, that virus might be attaching to your DNA in a spot where it's disrupting an existing gene. Therefore, the presence of viruses can definitely cause changes in the structure of your DNA, and so certain viruses, so-called DNA viruses, can be possible sources of mutations. Now, last but not least, in terms of causes of mutation, there is something we call natural mutation. Now, natural mutation basically is scientists speak for hell if we know. That sometimes mutations happen with no evident external cause whatsoever. Sometimes mutations happen for no apparent reason. There seem to be just glitches in the process, which is unsurprising. Even a well-tuned machine does not work perfectly every time. And given that your DNA is 5 billion base pairs long, it is reasonable to expect that once in a while, an error is going to creep in. That's not really surprising. So sometimes there's just a glitch. The rate of natural mutation is actually fairly consistent across time and species. We can expect approximately 1 natural mutation for every 100 million DNA base pairs. That's about the rate of natural mutation that we expect, which means if you think about 5 billion base pairs in your genome, we can expect that the average person is walking around with somewhere on the order of 50 natural mutations in their DNA as compared to the genes that your parents passed on to you. So yeah, all of us are mutants. Every last one of us has mutated genes compared to our forebears. And natural mutation seems to be just a part of the evolutionary process. You know, it's a thing that happens periodically. Every organism is going to have natural mutations, and the natural mutations that happen to work out well are the thing that drives evolution forward. But we'll say more about that a little further into this lesson. I do want to point out, and hopefully you noticed this, that most of the causes of mutation that I talked about are completely natural things. We hear mutation and we assume, oh, it's because of some bad chemical that somebody put in our food, or it's because of dangerous radiation emitted by a human power plant or something like that. The overwhelming majority of mutations are natural mutations. That's the far and away most common cause of mutation. Furthermore, the majority of mutations caused by outside interference are still caused by natural phenomena. There are plenty of mutagens and carcinogens that occur in the natural environment around us. There are mutagens and carcinogens created by your own body. Formaldehyde is a byproduct of certain chemical reactions in your body, but it's in such tiny amounts that it's generally not a problem. But if it happens to build up around a cell or two, those cells or two could have gotten mutated by it. Radiation occurs in nature all the time. Ultraviolet rays. Viruses are natural phenomena. So the overwhelming majority of things that lead to mutations are not, in fact, things that people do. They're just things that we are exposed to as part of being alive and walking around in the world containing chemicals and radiation and viruses that can potentially get into our bodies and cause issues. All right, so we've seen a couple ways mutations can happen. What are the effects of mutation? Now, fundamentally, what mutation is doing is it is changing the structure of DNA. That is the basic thing that a mutation is accomplishing. It is modifying the structure of your DNA from whatever its base form was. Now, there are several ways that that can look. And so what I'm about to tell you about, you might say, are types of mutation in terms of ways that they can change the DNA. So in order to do this, I'm going to draw for you a couple of DNA sequences real quick here, and we are going to see how they can potentially be modified by different varieties of mutation. If you are following along with my handout, I've got examples of some of these presented as well, but I want something to hold up to the camera. Okay, here is a good base DNA sequence to start with. ATC, GGA, TAC. That's going to be our starting DNA sequence. Just pretend that's an entire gene. It's only 3 codons, and there are no genes in the real world that are only 3 codons long, but for the sake of argument. Now, the very simplest form of mutation is what we call a point mutation, also known as a substitution error. Here I have introduced a point mutation to my genetic sequence. You might see that the 4th nucleotide, as compared to our starting sequence, was changed from a G to a T. The wrong nucleotide ended up in that slot. That's what a point mutation means. It's a mutation that hits one point in the DNA. It changed exactly one nucleotide. Point mutations, or substitution errors, as we might call them, are the most common kind of mutation, and they are the ones that are most likely to slip by without causing any harm for the organism. Not to say that they can't. The original mutation long, long ago that caused sickle cell anemia, that has been inherited by people ever since, it was a point mutation. It was the difference between healthy DNA for hemoglobin and sickle cell gene for hemoglobin is exactly one nucleotide out of the 108 nucleotides in the hemoglobin molecule, or in the code for the hemoglobin molecule, I should say. So point mutations are the most common and the ones that might end up slipping by with no harmful effects at all. We'll talk about reasons why a mutation might not cause a problem a little bit further along here. So let's go with something a little more severe, or a little bit more potentially severe. This is what we call an inversion, or a transposition error. You see that what happened here is that I swapped the 3rd and 4th nucleotides in the sequence as compared to the original. Saw in our original sequence up top, it went ATC, GGA. I have swapped the positions of the first C and the first G, so now it says ATG, CGA. That's an inversion. We flipped around a portion of the DNA so that a little bit of it reads backwards. Now you saw why that's potentially more serious, because in that particular example that I showed you, that inversion affected 2 codons rather than 1. A point mutation at most affects the code for a single codon, and that can be serious, as you've already seen. But that one affected 2 codons at the same time, which means the likelihood of it doing something that's going to disrupt the final protein that gene is coding for is much higher, because we've now just potentially changed 2 of the amino acids in the sequence that gene is giving us the instructions on how to build. Furthermore, what I just showed you was a simple inversion. That was an inversion involving only 2 nucleotides flipped around. Let's imagine we have a much more serious inversion, where I'm flipping around, say, 4 nucleotides, big chunk of the DNA all at once. Or maybe I'm flipping 9 nucleotides. Maybe I'm taking this entire sequence here and flipping it backwards, running it the other way around. There's no upward limit on how many nucleotides can end up being flipped backwards when you do an inversion. And if you've got an inversion that flips 9, 10, 15, 20, 50 nucleotides backwards, needless to say, you're disrupting a very significant portion of the gene. So inversions vary in severity depending on where they hit and on how many nucleotides are involved. If you're lucky, a small inversion that flips 2 nucleotides inside the same codon might not even have any noticeable effects on the organism. But if you're unlucky, then you might have a bad inversion that completely disrupts the gene. All right, let's get more serious still. While I sketch this out for y'all. Alrighty. Here is what we call a translocation error. In a translocation error, a portion of the DNA is removed from the sequence and reinserted in the wrong spot. Look at our original sequence up here that says ATC, GGA, TAC. Now look at the modified translocation sequence at the bottom. You see what happened is we took the 4th through 6th nucleotides in the original, CGGA, clipped them out, and reinserted them at the end of the gene. Everything that was between that point and where it was reinserted got messed up. You could see that when we did that, the TAC, the codon at the end, got bumped down a few letters and combined with the AT that were left at the beginning. And that's why we now have a thing that says ATT, ACC, GGA, because the CGGA got stitched onto the end and everything after it got bumped down to join with the part at the beginning. Needless to say, translocation errors get more severe depending on how far they travel in the genome. Obviously, the further you move along, the worse my translocation error becomes, because I'm pretty much messing up everything between the point where the fragment was removed and where it gets reinserted. The entire gene in between those 2 spots is going to be screwed up. It's potentially even screw up multiple genes at once if the fragment that moved into another gene. Now, you also are going to have this being worse depending on how big the fragment that moves in. You're probably going to have more problems from moving 12 letters than from moving 1 letter. You also, well, actually, maybe not. We'll talk about that in a moment. And finally, you're also going to have the possibility that when it moves, you can get an inversion at the same time as a translocation. Sometimes you move a bit, but that bit flips around before it gets reinserted, and you end up doing inversion and translocation simultaneously, which just further screws things up. So translocation errors are quite likely to cause some sort of issues. Now that said, I've not shown you the most disruptive mutations altogether. And this is a whole group of mutations that we call frame shift mutations. A frame shift error is going to be one that changes the number of nucleotides in the sequence. I'm going to redraw the original sequence at the top here, just to make things clear what is going on when I show you the ways that this can manifest. And then I'm going to show you a couple of ways that frame shift can change it. Okay. Redrew my original sequence right underneath where it says frame shift, ATC, GGA, TAC. Now, next to that, we have an insertion. In an insertion, we add one or more nucleotides to the sequence. You can see that what's happened here is we have an insertion in which two nucleotides have been added right after the first codon. We added an A and a C that weren't there before. And you see what happened, right? That bumped everything down. It changed every single codon in that gene from that point onwards. So adding even one extra nucleotide to the sequence basically messes up the entire gene going from that point forward because it shifts everything down one letter. It changes the reading frame, as we call it, of the ribosome when it goes to translate that gene, and therefore every gene from that point onward, it's shifted one letter down, and therefore every codon is going to be messed up from the point of insertion forward. An insertion could be a single nucleotide, an insertion could be a dozen nucleotides. It does not matter how many you're inserting. Either way, you're bumping everything further down and therefore changing the entire structure of the protein that is being coded for. All right, next possible way that a frame shift can work is a deletion. Remember our original sequence, ATC, GGA, TAC. In a deletion, we remove one or more nucleotides from the sequence. So you can see here in our deletion error, we've taken out the C that was originally in the third position. That causes everything to shift one spot, but it shifts backwards rather than forwards. But the effect is the same. It changes every codon from the point of error onward by changing the reading frame. You can see that we took out that C, and every other letter in the sequence got bumped back. Incidentally, notice how both the insertion and the deletion resulted in us having a couple of letters, that AC dangling at the end in both of these cases, that are no longer part of any codon. Those letters have been taken out of the sequence effectively. And as a result of that, they're just sitting in the DNA getting copied along with the rest of the DNA, but they're no longer part of a code for anything. They got bumped out of the gene altogether. This is a possible source of introns, folks. We believe that a lot of introns may have originated as basically leftovers from old mutations, where they're letters that got kicked out of a functioning gene but are still being copied along with the rest of the DNA, because DNA is really good at faithfully copying everything that's there, whether it's useful or not. And finally, when we're talking about frame shifts, one last kind of frame shift. I have not moved. How did I lose my pen sitting still in one spot? Here we go. All right, and our last possible frame shift that I want to introduce here here is what we call a duplication error, otherwise known as a trinucleotide repeat. In a duplication error, some portion of the DNA was read more than once. Basically, we kind of have like a skipped record effect, if you will. Old school record players where sometimes the needle on the record would skip and it would play the same bit of a song more than once. But that's kind of what's happening here, that we've repeated a portion of the sequence, maybe once, maybe several times. Now that portion could be one letter. We could have a duplication error in which we just go A, T, C, 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 and then move on to the next codon. But more often we see what's called a trinucleotide repeat, in which an entire codon or trinucleotide, 3 nucleotides, gets repeated more than once. And that, of course, is going to affect the final structure of the protein that we are coding for, because it's going to mean multiple copies of the same amino acid being inserted into the resultant protein. Interesting thing about trinucleotide repeats is that sometimes they don't have a lot of effect at low levels. Sometimes that repeats, so, okay, an extra copy of that amino acid does not meaningfully mess up the protein that we get, but you add another copy and maybe you might begin to have a problem, and you add another copy and you might have even more problems. We call this genetic anticipation. If we look at genetic anticipation, that means that we're seeing a situation with trinucleotide repeats in which the error is not yet severe enough to cause a problem, but we can see it's trending that way, and with subsequent mutational events, it could potentially end up manifesting in a dangerous form once you get enough of the repetition built up. We also find that certain genetic disorders vary in severity depending on how many copies of the trinucleotide you end up with. A classic example of this is a disorder called Marfan syndrome, capital M-A-R-F-A-N. Marfan syndrome is a disorder involving the formation of connective tissue in your body that causes your body's connective tissues to be formed incorrectly. As a result, you're much more prone to joint injuries and bone injuries. You tend to have a very long, lanky, narrow frame, kind of a scarecrow figure in a lot of cases, long arms, long legs, tall and narrow, high narrow face, that sort of thing. Folks with Marfan syndrome are way more likely to get, again, cartilage and bone disorders and breaks. They're way more likely to experience degeneration of connective tissues over time. And Marfan syndrome has varying levels of severity. At low levels of repetition of the trinucleotide, it might just mean that you have a slightly tall, narrow body frame, At high levels, you might experience severe connective tissue breakdowns that results in you dying quite young. There are a lot of possible ways Marfan can go depending on the severity of the repeats. By the way, you all, if you're kind of wondering, what am I talking about with that, like scarecrow, narrow face, narrow body kind of appearance, you all know someone who has Marfan syndrome, or more to the point, you know of someone who has Marfan syndrome. Some of you may have a portrait of someone with Marfan syndrome in your wallet right now because his name was Abraham Lincoln. And incidentally, Marfan syndrome, for reasons we are not clear on, is also strongly associated with bipolar disorder, which Lincoln suffered from throughout his life. So, well, we didn't know that then. It's kind of retroactive diagnosis. We didn't, neither Marfan syndrome nor clinical depression or bipolar disorder were known about in Lincoln's time. But looking back at his appearance and looking back at his biography, it is pretty evident what he was experiencing. All right, so those are all ways that I've said that mutations can look. Those are all ways that mutations can physically affect the structure of the DNA. Now, I mentioned a couple genetic disorders in there, and I want to point out an important thing about this, that like I said, okay, this mutation right here, there's a point mutation that's responsible for sickle cell anemia, or there's a trinucleotide repeat that's responsible for Marfan syndrome. A thing that you need to know about this is that most people getting a genetic disorder like that, the mutation did not just happen in them. The mutation did not happen just in them. The mutation did not happen in their parents. The mutation did not happen in their grandparents. Odds are that mutation happened many, 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 many generations ago. It doesn't have to occur newly in you for you to get that disorder. Basically, what this means is that somewhere in the distant genetic past, some ancestor had that mutation. That mutation changed their DNA, and the mutated gene has been getting passed on in the population ever since, because everybody who inherits that gene can potentially pass it on to their kids, who can pass it on to their kids, who can pass it to their kids. In other words, if you have a genetic disorder, odds are pretty good that it's not because a brand new mutation just happened in you that just happened to make it work out the same way that you end up with asthma or whatever. Odds are pretty good that you're simply inheriting a gene that the mutation for it occurred 1,000, 5,000 years ago, and is still just being transmitted from ancestor to descendant ever since. All right, now this brings us up to the next statement, or the next thing to consider here, I should say, which is the consequences of mutation. What does a mutation end up meaning for the organism that gets it? There are a few things that can happen here. There are a few ways that a mutation can end up changing the resultant's DNA. We can have what is called a missense mutation, M-I-S-S-E-N-S-E. A missense mutation changes the amino acid sequence of the resultant protein. In other words, we monkeyed with the instructions present in the DNA, and by changing the instructions, we now code for a protein that is different than it was before. A missense mutation, for example, is responsible for sickle cell anemia, because by changing one nucleotide for another, we now changed which amino acid that codon codes for, and the resultant protein that we produce, the hemoglobin, does not have the right formula. So that's a missense mutation. Another possibility is a thing called a nonsense mutation. A nonsense mutation causes a situation in which you've got a change that caused a stop codon to occur before it should. Remember, every gene ends with a stop codon that tells the ribosome, here's where you stop reading. But let's suppose we have a mutation that changes the structure of the DNA in such a way that now there's a stop codon in the middle of the gene. What that means is that anytime your body tries to transcribe and translate that gene, it's going to cut it short. It's going to end before we're done because the stop codon is going to artificially tell your cells, hey, okay, this one's finished. So that's a nonsense mutation, introducing a stop codon earlier. There's even a thing that can happen, and this is an interesting one, called a back mutation. A back mutation undoes the effects of a previous mutation. It's sometimes called a reversion, R-E-V-E-R-S-I-O-N. And a reversion, it basically ends up being a mutation that causes another mutation to go away. It's a mutation that undoes the effects of a previous mutational event. Not common, but it happens. All right. Now, what are the consequences for you, the organism? There was the consequence for the gene I just told you about. I just told you those three things, missense, nonsense, and inversion, are, by the way, inversions are also called back mutations. Not reversions. Okay. I think I mixed something up there. Not inversion, reversion. Reversion is the same thing as a back mutation, which means that, it's changing something back to the way it used to be. Inversion is where the part of the DNA gets flipped. So my bad, sorry for the confusing terminology. All right, so those are all things, back mutation, missense, or nonsense, that tell us what happens to the gene as a consequence of the mutation event. Now we need to ask ourselves about what happens to you, the organism, as a consequence of that mutational event. What does that mean for you personally? Well, a couple different possibilities here. And it really all depends on, first off, what cell the mutation occurred in, and secondly, on what exactly the mutation did to that gene. How severe is the damage? This is going to determine what happens next. But you know what? I have gone for approximately the length of a normal lesson right now, so I think this is a good time to cut short. I will tie this off. Next time we get back together, we're going to talk about the consequences of mutation, and one of those consequences is a thing called cancer. So we will get into that in our next lesson. I hope you are all having a good afternoon. Talk to you soon.